Welcome back. This is Mr. Hyatt talking about the bottleneck effect. We left off talking about the founder effect. I said that it's really similar to the bottleneck effect because we're taking individuals from a larger gene pool and we're shrinking that down to a really small size. So let's talk through the pictures first. So this is kind of a classic example. Notice that in the original population, we've got red. We've got blue, we've got white, and we've got yellow beads, or whatever those things are. There's some sort of a bottlenecking event. Notice it's called a bottleneck because old school bottles and really new bottles used to taper a lot at the top. Think of an old school Coke bottle, and so we can have a lot more diversity here at the fat bottom than we can at the skinny top. So something happens. Maybe they're being overhunted, maybe their climate is being changed, or their habitat's being destroyed, or something along those lines causes a large population to shrink to just a few individuals. Now look at our variability. We've lost all of the yellow trait, and we're down to 1 out of 2, 4, 6. 1 out of 6 white alleles, where that was not the original ratio. I could sit here and count all those up, but who's got time for that? Our ratio has been thrown off, right? So we've lost diversity. If that happens for a long amount of time, then we can see some really damaging things. We've seen what's happened in cheetahs because of overhunting. Dalmatians have some genetic defects because we had a low population size. And then all of a sudden 101 Dalmatians came out and every little kid wanted a Dalmatian. So we saw the opposite of the bottleneck. So the population came out of a bottleneck and rapidly expanded, and we saw a spread of recessive alleles. The book talks about the impact of genetic drift on the greater prairie chicken. Notice that the loss of habitat caused a huge reduction in their numbers, specifically in Illinois. The surviving birds had low levels of genetic variation, and only 50% of their eggs hatched. Think back to when I talked about the fact that humans today, males, produce 50% of the testosterone of what our grandfathers did. So a really similar kind of phenomenon. So here we're seeing greater prairie chickens in Illinois pre-bottleneck. We're seeing Kansas, no bottleneck, and Nebraska, no bottleneck. Population size was between 1,000 and 25,000, depending on which of these 30 years we're talking about. But in 93, we were down to fewer than 50, or less than 50, whichever the correct way to say that is. Check out our number of alleles per locus. Remember, a locus is like an address on a chromosome, so it's a gene's address. So the number of alleles per locus dropped. And if we use these other groups to compare, we're drastically lower than those other groups. Percentage of eggs hatched, 93, 99, 96, less than 50 after the bottleneck. So it produced some real issues in these organisms. If we looked at sperm fertility and how many of our sperm relatively produce babies, I wouldn't be surprised if we see that same phenomenon in humans today. So what scientists did is they used DNA from museum specimens to come up with this data up here. And then they compared it to the other populations. The results showed a loss of alleles. I think we're pretty good there. So just to summarize, genetic drift is significant, especially in small populations. If you've got a large population and you drift a little bit, no big deal. If you have a small population, it can cause really big issues. It can cause allele frequencies to change, lead to loss of genetic variation, and it can cause harmful alleles to be fixed. Think back to the flowers. If that red allele is bad, everybody is screwed. Gene flow is real easy, immigration, emigration. Maybe we're talking about between two populations. Remember, population means same species, same place, same time. So maybe we're talking about a population of birds that live on opposite sides of a mountain. One day there's a tornado that blows a bird to the other side of the mountain and introduces a whole bunch of different traits. Same species, different population. Gene flow can definitely affect adaptation to local environments. It's going to inject a whole bunch of new alleles that haven't been found in that population before. We see a specific example of this in the banding pattern of Lake Erie water snakes, and I've got a picture on the next slide that's going to show that. But notice before we head to the pictures that most of the snakes on the mainland are strongly banded. Most of the island snakes don't have bands, or they're somewhere in between. Unbanding makes you a better camouflage, or makes you better camouflage in the island habitat, so you fit your environment better. Remember, survival, success, and mate choice. Migration allows for that banded population to show up sometimes in the island population. So this area, it's a ferry ride from here near Sandusky out to Kelly's Island. I've been to this area a couple times. But notice that on land we've got banded, on the island we've got mostly non-banded, but we do have some snakes that migrate out, and they catch a ride on the ferry, or somebody brings them out, something along those lines. And there's kind of a graph again that shows those banding patterns. Gene flow can increase the fitness of a population. It can also save a population. Like the greater prairie chickens, we introduced prairie chickens from Nebraska and from some of those other states, and we were able to kind of recover some of the genetic diversity, and we were able to increase the egg hatching rates. So consider the spread of alleles for resistance to insecticides. That's something we talked about briefly in the last chapter, but I said we were going to get to it more in this chapter. Notice that we've used a ton of insecticides, DDT, and lots of derivatives of DDT, to target mosquitoes that carry West Nile and malaria. So in some populations, those mosquitoes are resistant to those insecticides. If that population then spreads to another population, maybe a mosquito jumps on a plane and hitches a ride to the U.S., now that insecticide resistance is going to be present in our population. An important agent of evolutionary change in modern human populations. Think about how interconnected our planet is, how much more global travel there is today than there was even 50 years ago. So we get a lot of alleles coming into our population that maybe originated in Northern Europe, or maybe originated in Southern Africa or Asia somewhere. Moving on to 23.4. Natural selection is the only mechanism that we can identify that consistently causes that adaptive evolution where we get better and better as time progresses. Natural selection involves both chance and sorting. We definitely get new alleles from genetic variations, and those beneficial alleles are going to be sorted out and favored by natural selection. I'm sorry, I kind of lost my train of thought there in the middle. So I left this slide in. I wanted to make sure that I talked about the fact that evolution is not goal-oriented. We aren't the point of evolution, but evolution doesn't act on genotypes. It doesn't matter whether you're a heterozygote or a homozygote. Are you tall or are you short? The phenotype is what matters in natural selection, not the genotype. So sometimes recessive alleles can stick around because they hitch a ride with a dominant allele. So relative fitness is what this is all about. Survival of the fittest, I really don't like as a teacher. The struggle for existence, I don't like as a teacher, because it does imply that if there's a fittest, there must be a less fit. So somebody must lose. Reproductive success is almost always more subtle and depends on many factors. So your relative fitness is your contribution to the next generation's gene pool. I've talked a little bit about this with replacement fertility rates and those types of things. My sister currently has a higher fitness than I do. She has 3 kids. I have 2 kids. The next generation has more of her alleles and her husband's alleles than they have of mine and my wife's. So they're relatively more fit than I am. You maybe talked about this as freshmen. We need to be able to discuss like real life examples, and we need to be able to look at the graphs. We need to identify what type of selection are we seeing? What type of evolution are we seeing? In directional selection, you see here, things are moving in one generation. Moving in one direction. The example that I'm going to use is fur color. So the original population is here. This must be a dark habitat because it's better to be dark than it is to be light. So here we're shifting towards dark. We're shifting our population distribution towards dark. So we're selecting in one direction. We're evolving in one direction. Disruptive selection would be where the way I like to describe this is the freaks take over. It's good to be white or it's good to be really dark. In the middle, you stand out. So maybe we've got a habitat where there's some sand and some rocks, some really white sand. So these guys are camouflaged, these guys are camouflaged. If you're in the middle, you stick out. So the population becomes white and black with no in between. Stabilizing selection is the opposite, where the middle is best. Think of like human blood pressure. It's not good to be low, it's not good to be high, it's good to be right in the middle. So how does natural selection impact adaptive evolution? We see octopuses that can change color rapidly for camouflage, things like not iguanas, chameleons that can change color. Pretty striking adaptations that we've seen because of natural selection. Another example that your book talks about that I'm going to do a really poor job of explaining is how we have these movable jawbones in snakes, and some other organisms. They can basically detach their jawbone and reattach it to swallow things much larger than themselves. That's an odd adaptation. Make sure you look at how that developed in your textbook. So the key role, continued. Natural selection is going to increase the frequencies of these alleles. If you're fit, your genes get passed on. So that should mean that good genes are getting passed on, which means our next generation should be more fit to their environment than the current generation. But if the environment changes, adaptive evolution has to change. We have to adapt differently. Genetic drift and gene flow sometimes go back and forth. And sometimes they don't enhance survival and reproduction. Sometimes they're going to be negative mutations. Sometimes we're going to lose some positive traits, again, through genetic drift. Maybe our best organism flies away and goes to another population. Our most fit organism goes somewhere else. And we lose a lot of those really adaptive and beneficial traits. Sexual selection is going to be where you're more attractive to a mate. So I'll post a link to Birds of Paradise mating dance. It's those types of things can result in sexual dimorphism. Here's an example of that with a male and a female peacock. So it's the female is over here. The male has to show off to get a mate. So our coloration patterns get really crazy. If you're a bird of paradise, your mating dance gets really crazy, gets really intricate, and your coloration pattern gets really unbelievably crazy. In terms of sexual selection, we've got intra and inter. Anytime you're talking about intra, that's within. So we're talking about mates competing for a mate within one species. So that's maybe what high school feels like for some of you. A lot of people competing in different ways for mates. Intersexual selection is where the individual of one sex is choosy in selecting their mates. That's what we see a ton. They choose. The female chooses what the desirable trait is. I apologize, I'm stumbling through this. I lost my train of thought in the middle of that again. I'll pick up with sexual selection in video 3.